Hello everyone, my name is Xin Liu. I'm a researcher and engineer in the Massachusetts Institute of Technology.
Hello, my name is Pat. I'm also a researcher at MIT. We present Micropad, Enhancing Microbial Consortium for Plastic Biodegradation. We aim to identify the consortium of microorganisms that can efficiently degrade polyethylene or PET waste and produce high-value plastic monomer BKA, a precursor for nylon. This process is known as upcycling, using the unique environmental stressors in the International Space Station, including microgravity and radiation, as selective pressures and catalysts for the sustained enhanced bioactivity. Motivation for the project. Our prior work on Micropad 1 identifies Pseudomonas putida, a microorganism able to break down polyethylene and upcycle it into BKA, a performance advantage bioproduct that can be polymerized into nylon. Our collaborators at the National Renewable Energy Laboratory, or Ariel, has a long-standing experience in large-scale manufacturing of plastic upcycling applications. Our output of the experiment provides a sample and data on high-yield microorganisms that can directly be integrated into the ground development in Ariel enzymatic PET depolymerization platform. For the hypothesis and research question of this proposal, we hypothesize that a consortium of microorganisms, including Pseudomonas putida and other microbes, can improve the efficiency in the degradation of plastic waste and production of higher value plastic materials compound compared to a single-strain microorganism condition. Further, we hypothesize that the microgravity and radiation of space can serve as the selective pressure that could catalyze and sustain the enhanced upcycling activity once the microbe return to Earth. For the rationale of using the International Space Station, research has shown that stress response caused by microgravity has induced microbes to have enhanced bioactivity that allows the cell to have a higher metabolic rate. Microgravity also changes the microenvironment of the organisms due to lack of buoyancy, permitting study, selection, and manipulation of how microorganisms acquire nutrients from their surroundings. Finally, radiation may increase microbial mutation rates during the flight, leading to the potential adaptation and coevolution of the microbial community. Thus, it is promising to use the unique condition of the International Space Station National Lab to incubate and cultivate strains of microorganisms with beneficial capabilities.
In order to do that, our automatic programmable culture payload will proceed with the pre-programmed experiment once in orbit, in which we limit carbon source in the growth medium, providing selective pressure for evolution towards activity and growth of the microbe that degrades the PET more efficiently. Currently, we are working on the first iteration of the experiment that is scheduled to launch on board SpaceX 25 inside the NanoRack black box, in which a single strain will be tested out both for the hardware and our in-orbit operation in a full hands-off setup. We already identified our optimal in-orbit logistics and the potential risks, such as failures of the hardware and unsuccessful preservation of the biological samples pre- and post-flight. However, with our current work as the technology demonstration, we will have the expertise for handling the risk. More importantly, our prior work with Ariel on the enzyme and microbes, and our collaboration with Chris Sanders' lab in Harvard, Seed Health Research Lab, and Professor Christopher Mason in Cornell to ensure the success of the project. And after the flight, we will be measuring the biodegradation results through physical analysis and chemical analysis. We're looking at quantitative genomic, metabolic, and proteomic analysis to understand the microbe adaptation and biodegradation pathways.
There are multiple outcomes and benefits for the project. For the scientific and technical benefits, quantitative genomic, metabolic, and proteomic analysis will lead to the understanding of microbial adaptation, biodegradation, and upcycling pathway through high-quality validated genome-scale models. For the economic and commercial benefits, the global market for plastic recycling is projected to reach $47.3 billion by 2026. The upcycling process in this mission creates valuable products from PET and ultimately incentivize higher reclamation rate at scale. Through our partnership with the National Renewable Energy Laboratory, or Ariel, and MIT entrepreneurial ecosystem, the technology developed in this project could translate to sustainable market and commercialization. For the social and humankind benefit, a higher yield and upcycling process will create a circular economy that extends the functional lifespan of molecules to make plastic and reduce waste. Finally, for the STEM benefits, we plan to create public exhibitions for the project, as well as using social media outreach, leveraging MIT resources to generate interest in space research and raise awareness for sustainable development in schools and public.
Here is our cost estimate and our schedule. We're very much looking forward for the support from ISS National Laboratory. Thank you so much.